All right. Okay, thanks everybody for coming out tonight. This is our first Boston Protein Design and Modeling Club meetup of 2025. So thanks for coming out. I know that way back in 2024, I stood here and I told you that was our last meeting in this room, and it was a bald-faced lie. This room is going to be available to us until about June. My department can't use this room because construction is actually going on, not on this room, but near this room during the day. And so during nighttime, there's no construction. We can use this room. Okay. What's that? Google also says it's permanently closed. Don't listen to Google. It's open. For us and for us only. That's for everybody else. That's for everybody else. Okay, so we've got a great speaker tonight. I'm a little biased because Ben Fry is actually a graduate student in my own lab at HMS and at Dana-Farber. So a little bit about Ben. Ben is a third year biophysics PhD student at HMS. And he is a NSF graduate research fellow, just got the NSF fellowship this year. Before he joined Harvard for grad school, he was an undergrad at Johns Hopkins. He was actually spent some time as a Rosetta REU student working in a group at the Wistar Institute, where he was doing computational modeling of glycoproteins, of the glycosylation sites of glycoproteins. He continued that work at Hopkins in Jeff Gray's group. And then when he started at Harvard, he rotated in my group, and I thought, wow, I got to make sure I can do what I can to keep Ben, because he's going to be a major star in the protein design community. And I think that the work he's going to talk about today shows that that's true. So this, as an aside, I gave a short talk at MIT about Ben's work back in November, and people came up to me after the talk and they said, "Is Ben on the job market?" And I was like, "Well, he's third year. No, I don't try to steal him from me right now." And okay, because the way you were talking about Ben, it just seemed like you were trying to, make him really look good for the job market. I was like, no, he's just really good. So this work. So Ben is purely computational in my group. Maybe we'll get him pipetting someday, but for now he's doing really good at burning through GPUs. But the work he's going to talk about is a very big collaboration with a research tech in our group who's doing a lot of the experiments, Kaya Slaw, who's actually here tonight. So a lot of the work of the experimental interrogation of what Ben is going to talk about is actually done by Kaya. And so with that, I'll step down. And well, first I want to thank again Twist for donating to the Protein Design and Modeling Club, so we could have this nice pizza and drink our nice drinks. And if anybody else wants to, any of the pharma or biotech listening or on YouTube, if you want to support us, just email me. We can figure, there's actually a link on our website to do so. But without further ado, I'll give the reins to Ben to tell us about his exciting work. Thanks, Ben.
All right, thank you everyone for being here tonight. Can everyone hear me? Is the microphone good? And Zoom, can we get some thumbs up maybe just to make sure? Yeah, like Nick said, I'm going to talk about what I've been working on for the past basically 2.5 years, I guess, in the Polizzi lab, designing small molecule binding proteins with deep learning methods. So if we want to create a protein binder to an arbitrary small molecule ligand, there's basically 3 ways or 3 subproblems that you need to solve. The first of which is to choose a fold that you're going to try to bind the small molecule with, or a scaffold. The second is to stick a ligand into that fold to sort of choose the placement that you want to go after for sequence design. And then finally, to actually design the sequence conditioned on the three-dimensional structure and the placement of the ligand inside of it. What I'm going to present today is our model for doing this third task, designing the sequence conditioned on full atomic structures, which we call LASErMPNN, which stands for ligand-aware sequence engineering message passing neural network. And as an aside, which I'm sure many of you are aware of, this is very similar to a similar model called ligand-MPNN, and I will do my best to draw comparisons to that model. But sort of to motivate why we have developed this model in the first place, when we started this project, both AlphaFold and Protein-MPNN could not handle ligand information. So a motivating question for us when we started was, how can we from scratch create a model which generates sequences from protein structure while also taking ligands into account? And furthermore, can the deep learning methods that we develop actually increase design success rates or affinities over previous methods? And I'm going to start by talking about the first of these questions. So if you want to train a deep neural network to generate ligand binding protein sequences, the first thing you need to do is start with the PDB, as all good protein design deep learning models do. And what we're going to do is we're going to take every single molecular interaction as an example, which we can learn from. So this might be a protein ligand complex, it might be a protein-protein interaction, and it might be protein nucleic interactions. So taking one of these as an example, we're going to strip off any native sequence and rotamers. And then we're going to take this quote unquote stripped backbone and feed it into our model. The output of which is going to be a probability distribution over the 20 different amino acids. And if we sort of zoom into like an example crystal structure, a glutamine specifically, you can see that the model outputs amino acid probabilities corresponding to sort of similar polar things that you might expect to find on a protein surface. When we start training this model, the predictions are very like uniform across all of the amino acids, but over many iterations of training, this sort of converges towards more confident predictions. Oops. Yeah, converging towards more confident predictions. So the objective in the loss function for this model is a cross entropy over the correct amino acid prediction, as well as over the side chain dihedral angles for our model specifically. We also have a small term that encourages the outputs, the probability distribution, to look like MSA statistics as well, which I should mention as an aside. So if we want to generate protein sequences from this model, we can use the generated probability distributions and sample from them, placing amino acids with probabilities proportional to what the model predicts. So for example, at a site that's on the surface of this protein, you might put an arginine, you might put a serine to cap your helix, but it's a little bit less likely to put something like an isoleucine here. For our model specifically, again, once we sample at one position, we're going to fix that sequence and rotamer and then hop somewhere else in the protein structure before designing. So very similar to protein and ligand-MPNN. And just like protein and ligand-MPNN, we have this sample temperature knob that we can turn in order to trade off the confidence of the model for sample diversity. And this is an important point that we'll come back to later. But essentially, we can get we want to balance picking a temperature that's high enough to get lots of diverse sequences, get a lot from, also ensuring that the sequences we're generating are still folding to the 3D structure that we want to fold to and potentially binding our ligand. So I'm going to show a block diagram of the architecture that we've developed for this task. And this is a lot. So I'm going to start by zooming into one small part of it. And that's going to be the small molecule encoder, which is tasked with building up useful representations for each atom in a small molecule that we want to bind. So again, if our goal is to build up these useful representations, what we want to do is find a data set that has more diverse interactions or more diverse ligands than potentially the PDB does. Because we know sort of the types of ligands that exist in the PDB might be limited relative to what we can find elsewhere. So in specific, we're going to take the SPICE dataset, which has over 100,000 unique small molecule atomic systems in many millions of conformations, and also has, for every single atom, partial charges and dipole vectors from density functional theory calculations. And from these molecules in this data set, we can also predict connectivity-derived features such as formal charges, numbers of covalently attached hydrogens, atomic hybridizations, and aromaticity. And the goal of this all is that we're building up useful representations that we can feed into the encoder and decoder trunks of the LASErMPNN architecture. So a key point, a key decision that we made in this process is actually taking the predictions of all of these features from the small molecule encoder and actually freezing them during the training of the sequence design model. And something that this allows us to do is when we use this model for inference, actually interrogating its predictions of the connectivity-derived features, the quantum mechanical features, to test if the model understands the small molecule that we're trying to bind. So for example, I'm showing you biotin, which was not trained on in the SPICE data set, and the models predicted, or the ligand encoders predicted partial charges per atom. And this looks reasonable if you'll trust my word for it. Something else that's sort of interesting is if you actually don't pre-train this ligand encoder before training the full LASErMPNN architecture, the accuracy goes down. So the diversity of the data in the PDB is not actually as useful as what you might find mining the SPICE data set, which is pretty interesting.
How meaningful is a 3% difference?
I would say, oh, sorry, the question is, how meaningful is a 3% difference? And when you train a lot of these models, you're typically looking at like plus or minus 1 or 2% between different replicates of like the same like hyperparameter settings, right? So I would say in this case, this is decently meaningful, but it's hard to say without doing lots of replicates, and this gets kind of training expensive when you have to like train many copies of them in order to get good confidence bounds. But I would say this definitely replicates in this case. There's other things that may not, as we'll get to. So moving on into the sort of encoder decoder blocks, which are tasked with building up representations of the protein, which eventually get decoded to both sequence and rotamers. We can sort of represent in this cartoon the heterograph formulation of how to represent our protein ligand system, right? And essentially what this is going to be a nearest neighbors graph composed of the ligand atoms, where every single node is featurized with representations built up from the small molecule encoder. And protein nodes, which are initially initialized to zeros with edges that encode the distance between everything. And from these distance sort of constraints, the model learns to map from three-dimensional structure to a distribution over probabilities. And I should mention the protein nodes are constructed at the level of NC alpha CO frames, whereas the ligand nodes are represented per atom. So it's sort of two different types of nodes. So if we zoom into the one-hop neighborhood of one of these protein nodes and look at sort of the different types of information that connects that protein node to other neighbors in the graph, our formulation for how to actually compute and build up useful representations is a play on the GATT-v2 architecture, which is from this Brody et al. 2022 paper. And essentially what this is going to do is we have two different sets of learnable parameters, so A and W in this case, and these are each going to be tasked with different types of pairwise interactions, right? So the blue ones might handle protein-protein interactions and the edge between them, while the green ones might handle protein-ligand interactions. And a key thing for this architecture is that we're going to softmax over the full neighborhood. So sort of respecting the protein-ligand graph as like full structural context in order to like pass structural information into a protein node. And from this softmaxing process, we're going to get attention coefficients, which we can use to weight the information that comes from protein MPNN style updates, which are also separated into protein-protein interactions and protein-ligand interactions. So sort of we're respecting this full protein-ligand heterograph in the message passing process. And to further illustrate the differences between this construction of our message passing against protein and ligand MPNN, if we take this diagram stolen from the Ingraham protein sequence generation paper and sort of extend it to represent what ligand MPNN is doing here, ligand information is handled strictly in the encoder. And there's 3 separate layers or rounds of ligand to protein encoding, followed by 3 rounds of protein to protein encoding, and then 3 rounds of sequence autoregressive generation or decoding. In our LASErMPNN architecture, we have 3 rounds of joint heterograph structure encoding, and then 3 rounds of joint heterograph structure decoding. So using this sort of HETCAT v2 architecture to do this. And what you see when you actually like compare these models, yes?
Yeah, there's no ligand-ligand interaction.
The ligand-ligand, so the question was, there's no ligand-ligand interactions, and that is not true. I'm not referencing it here because we have this ligand encoder that's pre-trained to build up those representations. So they're sort of fixed for our purposes. In ligand-MPNN, what's interesting is that the ligand graphs are actually fully connected in order to like build up their representations, which might help with like larger systems like large nucleic acids and stuff. But in our case, we use KNN graphs, so it's a little bit different. So when you compare sequence recovery from models trained with our architecture and you sort of ablate out all of the additional stuff that we've added in, and essentially what this blue box is showing is a model that's as close as possible to ligand-MPNN. It's only been trained to recover sequence similar to ligand-MPNN. These architectures are pretty similar, is pretty much the takeaway. On some replicates, maybe it's slightly better. But again, we sort of started this project before ligand-MPNN existed, so it's sort of interesting that we arrived at similar places. But we can increase performance by introducing extra features into our architecture, which I'll talk about in a bit. I should also mention or re-emphasize too that the diagram that I put up showing the decoding process is a bit, it's missing something, and it's that we are also decoding and conditioning our sequence generation on sampled dihedral side chain angles as well. You might be, if you've used ligand-MPNN, you might know that it uses or can generate 3-dimensional structures conditioned on the sequences that it's sampling. But the way that it does that actually is by using a separate trained packing model. So it's a completely different architecture that after the sequence is generated, builds the dihedral angles, all of the chi1s at once, all of the chi2s until it has a 3-dimensional protein complex. So ours is sort of contrasted in the sense that it's doing it 1 amino acid at a time rather than everything all at once. And part of the message passing formulation that we use to build up useful representations to do this rotamer generation task involves using geometric vector perceptron layers to build vector representations on all of the nodes, which enables the model to learn sort of relative orientation information. But I won't dwell on that too much. So sort of overall sequence recovery comparisons, if we take the ligand-MPNN weights that you can get and download from the authors and sort of compare them to our best full model with all of the features that we've added, the 2 models perform very similarly, which is interesting. But I will note that an exact comparison between these models is a little bit difficult because the ligand-MPNN training data is not publicly available. It's sort of released as a trained set of PDB codes and a test set of PDB codes, which makes doing an exact reconstruction of their training database to train them on the same things really difficult. So what I did to make this comparison is completely re-implement their architecture from scratch, which was really fun. Use the sequence clusters that we have for our own data set split and sort of hold out anything from their test set from our training set, any clusters from that. And what you see is that if you do an apples to apples comparison of LASErMPNN versus the ligand-MPNN architecture, we perform slightly better, which is interesting.
So if I understand correctly, so you know like which proteins were in like training, which were in test, but you don't have their training code?
No. So the question was, we know what proteins were in train, we know what proteins were in test, but we don't have their training code. And that's correct. What we have is the supplemental information from their paper, which is a big list of training PDBs and a big list of testing PDBs. There's some nuance to this, right? Because part of the training protocol is clustering the entire PDB at like 30% sequence similarity. But where you actually draw the boundaries between those clusters is going to differ depending on like what random seed you use in MMSeqs. Right, so making like an exact comparison between these models is a little bit difficult, motivating retraining the architectures.
Can I ask a question about training data on it? I've always wondered, when you're curating the data from PDB to use for training, would you have to remove any like crystal artifacts, molecules that were just in the crystal structure but maybe aren't biologically relevant?
Yeah, that's a great question. So the question was, to summarize, do we remove crystallographic artifacts from the training data? And in our case, we have a flag in our training code where we can turn it on or off because it is like documented in the header of the PDB files. I believe I've trained my models with them because it's kind of more information, and maybe that changes like the rotamers subtly in a way that the model can learn from. But I believe I've also trained models without them, and like the difference is hard to say if it's significant in replication. Any other questions? Jeffrey. The question was, what accounts for the difference between the red and the blue bar? And I would say we do not know. And we cannot know without access to the training code. So please release your training code. So if we take every single point in the reconstructed ligand-MPNN test set and plot them, plot the sequence recovery of each sort of test set complex against LASER and ligand-MPNN, we see that they're super correlated, which is interesting, but there is, especially for the binding sites, subtle differences, which is kind of interesting. So these models are learning the structure to sequence mapping in very similar ways, right? So they're obviously being trained on similar data since we get a correlated probability, but the difference between these models is sort of a motivating, interesting reason to maybe use both in your own sequence design journeys, I suppose. So the next question we wanted to assess is, when we compare these models, how do they do when they're wrong, right? So what we've done here is plotted the distributions of over the test set, the ligand-MPNN test set as reported for small molecules, the distribution of Blossom 62 substitutions over incorrect residues. So if you take all of the residues after generating 5 samples at 0 temperature for each model, take the set of binding site or overall amino acids that both models get wrong, what is the Blossom substitution score of the mistakes, right? And to motivate this, sort of like a serine to threonine, which are 2 very similar amino acids, substitute with a score of 1, a positive 1 in the Blossom 62 matrix, whereas something like an isoleucine to an arginine, which are very like chemically different, has a score of -3. And what we see is that LASER is maybe slightly better for this task. So it generates more biochemically similar substitutions in the case that it's wrong. And perhaps a reason for that is that we use this sort of MSA column average probability in the loss at a downweighted proportion relative to sequence recovery to try to guide the model to understand, if you're going to be wrong, at least make biochemically reasonable substitutions.
I was wondering, so when you said, if you go back to the previous slide comparing ligand-MPNN versus LASErMPNN, like for points where one's better than the other, is there some pattern to this? Like, is there, for some reason, one approach versus the other approach works for certain ligands or scaffolds? Or, because you mentioned that maybe somebody should try both. So I'm wondering how do you decide which one to try for which problem?
So the question was, is there a reason why these two models might disagree? And I think it's pretty difficult to consider all of the possibilities, I guess. But if you have suggestions for how to do so, we'd be interested in digging into that a little bit more. But I don't have a clean answer, I guess.
Like, is this completely random? Like, is it possible, like, if you just retrain LASErMPNN again, like, would it shift which is better or which is worse? Like, is it an architecture thing, or is it something how you're encoding the information?
So the question was, is this something that like sort of replicates between training runs, or is it like differences in like the architecture, the encoding? And I would say yes to all of it, probably, right? There's always going to be subtle differences between these models. I'm plotting the paper, the ligand-MPNN author distributed weights, which, as I mentioned, we don't really know what they did in terms of like what specific training data they have. So there are actually differences even in like what like training data cutoffs they might have used. So it's really hard to pin down like one specific reason, I guess.
It's kind of in a similar vein. Have you tried doing like two different trainings of the LASErMPNN, their sequence recovery against each other? It's like higher distribution or just the whole broad?
So the question was, have I trained multiple copies of the same LASErMPNN model and done this same plot? And I haven't plotted them against each other, but upon replication, the accuracy is pretty similar between like replicates. My guess is what you're going to see is just differences due to noise and like the order in which the data set is sampled, right? Because all of deep learning model training involves some inherent like randomness, right? But my guess is that they would also be highly correlated up to some noise. Any other questions? Okay, moving beyond just comparisons soon. So I guess just to conclude the last slide, this is sort of an interesting finding that maybe the LASErMPNN architecture is understanding a little bit more about the like physical reality of small molecule binding and how to make intelligent decisions for what to substitute for other residues. So moving on to our second sort of motivating question, can we use the LASErMPNN or a combination of deep learning methods in order to actually improve the design success rates or affinities over previous methods? And just to motivate this question a little bit more, in previous work from the lab, we've developed sort of this traditional method, which we call combs, to generate small molecule binding proteins. And this is a structural bioinformatics way based method to generate binding sites, which are then completed, fixed and completed using Rosetta, or you could use protein MPNN conditioned on those designed binding sites. And something that's really interesting is that when you use this method to develop binders to the small molecule apixaban, which is shown here, you can achieve a 33% success rate using this sort of traditional method with very high submicromolar affinity. And something that's interesting is that recent methods, such as Rosetta and ligand-MPNN, used to develop apixaban binders in a similar way, have a significantly lower success rate despite leveraging deep learning methods. And actually, like the highest affinity that was achieved in this Lee et al. Paper was actually higher or lower. It was a worse affinity than what was achieved using the traditional methods. So can our deep learning methods learn to actually improve affinities and hit rates over sort of our traditional methods, or can we use them in ways in order to actually do this? So I'm going to motivate this question by looking at a target for small molecule binding protein design, and this is exatecan. This is a small molecule, which is pretty interesting as a test of our model because it doesn't exist in the PDB or the Cambridge structural database. So we don't really know exactly what the conformer might look like, and structure prediction methods haven't been trained on it. It has structurally similar ligands. So I'm showing topotecan, which is related, but these structurally similar ligands actually intercalate DNA and bind into a DNA protein ligand complex. So if we wanted to develop a protein binder to these small molecules, we can't just take an existing natural protein and optimize that because we don't have a natural protein binding interface to go from that, to start from. So essentially what we're going to do in order to create scaffolds to pass into the ligand or the LASErMPNN workflow is take a large library of helical bundle backbones, which we love in our lab, and here they are colored by AlphaFold 2 PLDDT, so very confidently predicted over many different sequences. And we're going to use our Combs method to stick a ligand into these backbones. And what we're going to do is compare our sequence design methods for LASErMPNN versus the traditional workflow of Combs followed by Rosetta. And what we've developed in the lab is a so-called flexible backbone design loop, which leverages LASErMPNN, which predicts structure or predicts sequence from structure, as well as a structure prediction network, which predicts structure from sequence in order to subtly optimize the protein ligand binding pose over many iterations. So we're going to start with a single protein ligand backbone generated by the Combs protocol, pass that into LASErMPNN, generate 1,000 sequences at a decently high temperature to get diversity, and then pass that to Rosetta fold all atom. We can then take those outputs and filter them for several features of interest, and then pass those back into the LASErMPNN model to generate further sequences. Yes?
Do you actually need 35
For this, yes, especially for chiral. The question, sorry, was do you actually need 35 recycles for Rosetta fold all atom? And yes, for testing in our previous works, 35 recycles is sort of necessary to get the rucaparib helical bundle that we designed previously predicted correctly. So I would suggest using more modern AlphaFold 3 class prediction methods as they're much more computationally efficient. But at the time, Rosetta fold all atom is all we had. So again, we're using this. Oh, sorry.
So you do 35 cycles of the whole loop?
Yes. So there's 35. Sorry, finish your question. I'm sorry.
Yeah, no, that's the question. Like, what is the backbone RMSD between your starting helical bundle and your end helical bundle after doing that 35 times?
We'll get there. Okay. So yes, there's 35 recycles for Rosetta fold all atom, but there's also 35 iterations, and that's just sorted by random chance. But the key filters that we're going to use to evaluate these backbone candidates for new inputs to LASErMPNN are confidently predicted structures, low RMSDs to the previous iteration of design, and specifically for our case, we want to maintain that parts of the ligand remain buried, right? Because it's generally pretty planar, and we don't want to expose that to the solvent. And what we see is that if we plot sort of the PLDDT of the predictions versus the RMSD of the Rosetta fold prediction to what went into LASErMPNN for sequence design, over several iterations, this distribution shifts up and to the left, meaning that the outputs become more confidently predicted and lower RMSD, which is pretty cool. But sort of the key and the reason that we need to do this is because we need to subtly optimize from our starting coordinates. There are very small differences in ligand placement and backbone degrees of freedom that, like, to I look very similar, but to LASErMPNN and potentially Rosetta fold all atom are very important for prediction. So from this 35 iteration process, we've generated 100,000 sequences. From these sequences, we're going to filter them down. Again, we want high PLDDT, low self-consistency RMSD from the Rosetta fold predictions. We want the cores to be packed, so we use Rosetta to check that. And we also do a pre-organization check. And what this is in simple terms, is taking the apo and the holo structures of the ligand binding protein and repacking the apo structure, then superimposing the ligand back onto the binding site residues. And if heavy atoms are clashing after you repack the apo, we throw that design out. So we're sort of testing that the binding site is pre-organized even in its apo state. Once you do this, you have around 4,000 designs which pass your filters. And then if we look at those, we want to rank them for experimental characterization. So the ranking methodology that we've developed in the lab is to minimize the number of buried polar atoms that are not satisfied by a hydrogen bond. And what this might look like in an example hydrogen bond network is sort of forming lots of polar interactions, but not burying some sort of like charge thing in the core, right? So for example, this would be an example of something that's very bad because this is unlikely to actually fold or bind our small molecule. But what's interesting is that the structure prediction networks don't really seem to get this with high confidence, and you can still get confidently predicted folds that have buried charge residues in them. So by ranking our designs by minimizing the number of buried polar atoms, we sort of filter these terrible designs out. In order to select designs for experimental characterization, we outputted the top 10 and then chose 4 from that, which looked to our eye to be decent designs after following this buried polar atom ranking protocol. And what we saw is a 75% success rate, 3 out of 4 of our constructs bound, with a highest affinity of 0.3 or 300 nanomolar, 0.3 micromolar or 300 nanomolar, which was pretty cool. And when we compare this to our traditional method, Combs and Rosetta, both the highest affinity identified by this protocol and the success rate had improved, which was very interesting. Any questions?
I have a question about a couple slides ago. So you said you require them to be pre-organized. So is there any particular reason you're ruling out induced fit? Explicitly not looking for induced fit?
Yes. So the question is why prioritize and why heavily emphasize this binding site pre-organization and rule out induced fit as sort of a design target. And I would say that the induced fit model inherently requires some sort of energy to be put in that could lower your target affinity, right? So you have to pay potentially an energetic cost upfront, which maybe lowers energy eventually, but we want to develop things that are as simple as possible.
Well, it could also be just the proper temperatures, right? It could be for dynamic.
Yes, but dynamics is hard. Yes.
You said there's like a similar molecule in the PB. Did you check for like off-target binding to the other molecule?
The question is, did we check for off-target binding to like topotecan, which is that example, and we did. And I can show that in supplemental information later. And it doesn't bind, sorry. Or it binds the target with highest affinity and as a function of structural similarity of other molecules, less so.
You're talking about this small molecule that you don't have the conformer. For your training set, do you have a specific conformer for the small molecules? Do you have like a biologically relevant conformer that was bound to a protein, or it's just like RGT minimization of this?
So the question is, where did we get the conformer to do our design to? And I believe we took that topotecan molecule, which was structurally similar, and aligned a exatecan molecule onto it, so the actual thing we want to bind. And then we did QM optimization, so density functional theory calculations to optimize from there.
And follow on to the pre-organization question. So if it works right, so you're designing a 4-helix bundle with a hole in it, the apo.
Correct. The question is,
does it work?
the question is, does it still form a 4-helix bundle if it has a hole in it, right? And one of the nice properties of 4-helix bundles is that they're very designable. They can support many different sequences, and you can build a pocket into them. And I'm going to continue on for time, but essentially, we use this flexible backbone design protocol to develop binders with a higher success rate and higher affinity than our traditional methods. We solved their crystal structures. And what we saw is that the design model, which is shown in gray, to the crystal structure, which is shown in orange, is essentially indistinguishable when you superimpose them, which is really cool. We solved the structure at a very high resolution. So you can see the density of the ligand perfectly fits it like a glove, which is really cool as well. And the ligand RMSD is the ligand binds in the correct orientation with some subtle offset, but that subtle offset is sort of from the Rosetta fold-all atom prediction, which we can improve. And sort of zooming into that Rosetta fold-all atom prediction, this sort of provides us a fun opportunity to benchmark against the new class of structure prediction methods. So something that you notice from blindly applying Rosetta fold-all atom is that this tends to produce molecules with the wrong chirality. So this sort of flipping the amine over multiple replicates, maybe you get some that are right, some that are wrong. We can compare it to Boltz 1, which for some reason thinks that this ring is completely planar and aromatic, which it is not, and sort of problematic. Both methods are getting very low RMSDs, but if we compare it to AlphaFold 3, this one looks pretty good. So I'm sorry to the fully open source community. AlphaFold 3 still seems to be the winner in this case.
But it seems like if you put it in the center, it was right.
Mm.
He's like, you're not one or the other. No, it's not a question.
Moving on. So,
But I don't even know it.
Like if you can't decide between morality, that's what's good.
so sort of a question that we had is, can we use LASErMPNN to increase the affinity after we've already designed a protein, right? We use sort of a high sample temperature when we were generating these sequences, but can we do some sort of proofreading process where we go back, we look at the protein that we designed, and we sort of update what we think the sequence might be. And what you see is if you do this all but one binding site probability protocol where you take the entire protein sequence and its rotamers and freeze it, and then one by one you step through the binding site, and you ask the model, do you still think that this is the amino acid that should go at this site, conditioned on the rest of this sequence? What you see is that most of the binding site looks like this leucine 25 here, where the model is like, yeah, I designed this sequence, this is what goes here still. But what was really interesting is that if we take the model and apply it to this glutamine 51 residue, the model is now saying, oh, given this set of backbone coordinates and this placement of the ligand and the rest of this sequence, I actually prefer an asparagine at this site. And I might also prefer a serine. So this is also the case for this methionine 97 mutation. It much prefers a leucine at this site. So we made these mutations. And what was really cool that we observed is that the affinity of the Q51S mutant increased, but still sort of within the same order of magnitude. But the Q51N and the M97L mutants had actually improved another order of magnitude. So our takeaway from this is that this sort of proofreading process can recommend mutations which improve affinity, which was pretty cool in this case. One final interesting fact about this exatecan target is that it exists in a dynamic equilibrium between a bioactive and an inactive form. So this is a cancer therapeutic, which is often linked to antibody drug conjugates in order to target specific cells for cytotoxicity. But it's only active when the ring is closed, right? So what's interesting about this molecule too is that if you put it in aqueous solution at pH 7.4, the distribution of bioactive to inactive small molecule shifts almost entirely to the inactive form. But we've developed our protein to bind to the bioactive form, to the ring closed form. And we don't really see any evidence via molecular dynamic simulations or even just by looking at how things might work that the ring open or inactive form could even fit into the binding site. So you can monitor the relative proportion of bioactive to bioinactive forms of the small molecule by absorbance spectra. And if you look at sort of the peak to trough ratio at specific wavelengths, you can compute essentially how much ring closed or ring open form of your drug that you have. And what's interesting is if you take the drug in aqueous solution alone, sort of representing physiological conditions, the half-life of the ring closed form of the drug is around 2 hours. And again, we're monitoring that through this peak to trough ratio, which is what's plotted on this y-axis. But something that we hypothesized is now that we've added this designed protein, you can extend the thermodynamic cycle, right, to include the protein bound to ring closed form and the protein bound to ring open form. And again, we don't really see any evidence that the protein binds to the ring open form. So if you have a high KD or a very strong binder to the drug, you might be able to shift this equilibrium away from the ring open biologically inactive form. And what you see is if you do some kinetic modeling, if you assume that the protein binds to the ring closed and ring open forms equally, the half-life that you would calculate when you fit experimental data is an order of magnitude longer for the biologically active form of this drug. And if you take that point mutant identified by the proofreading process, this increases even further. But if we don't make this assumption that we bind the ring open and ring closed forms equally, and just represent this in terms of like the fraction from using like the dissociation constant to compute the fraction of the total ring closed form of this drug versus ring open, compared to the free drug in aqueous solution, we're going up an order of magnitude in terms of what might be bioavailable if you were to circulate this through a person, which is really interesting. So not only have we designed a really tight binder that exceeds sort of traditional methods, we've designed a function, which is rare for protein design. So sort of in summary, this protein that we've designed functions beyond just binding by stabilizing the bioactive form of this drug. Our model is competitive with ligand-MPNN for small molecule conditioned protein sequence generation, and combined with this flexible backbone loop, we're able to outperform our traditional methods. And with that, I will thank everyone for their attendance and their attention.
All right, further questions for Ben? I'm going to walk around with the microphone this time so he doesn't have to repeat the question. Jake.
So could you discuss a little more broadly why people would want to design proteins that bind small molecules? And also specific to your application, does the molecule need to be, apo, not bound to the protein in order to work and perform its anti-cancer properties?
So to answer the first part of the question, why would you want to do this, right? We generally think of like 3 different targets for this. Probably the most relevant to everyone is biosensors, right? So if you could have some sort of dimerization effect coupled to a fluorescence change, you could detect arbitrary metabolites for research purposes, or even like in situ in tissue. I'm happy to talk more about that, but sort of for the second question, do you mind repeating it or just prompting?
So if it is bound to the protein, can it perform its job that it's supposed to do once it's delivered to its target cell type?
Yes. So the question is, if the small molecule that we're trying to like distribute in our sort of potential drug delivery vehicle is bound to the drug delivery vehicle, will it be free to have its effect on the cells it's supposed to? And the answer in this specific case is that the small molecule is bound to antibody drug conjugates, which bind specifically to HER2 positive cells, right? Most of the tissues in the body are not HER2 positive, right? So what we actually want is to sequester that drug away from the rest of the tissue, right? We only want the drug in the place where the antibody is. And what might be possible is if that linker breaks, you can sequester that drug, right, and avoid some of like the off-target cytotoxicity that might result from specifically the ADC modality. And again, I can talk about this a lot more, but that's a one-point answer.
just to go back to that question, you still need to release the drug from the binder.
Yes.
So because this is not Ben's area of expertise, and because I'm here, I might as well step in. So yeah, I think the drug to bind topoisomerase 1 has to not be bound to the design protein. It's a non-covalent and reversible binding, and so if you couple this to targeting, you fuse this to some antibody or affibody that's going to target that protein, this design protein, to a cancer cell, for example. If it gets internalized, catabolized, the protein will get catabolized. They have some off rate of the drug, and the drug will be free to bind topoisomerase. So I'm not too worried about, it's not covalently linked, so I'm not too worried about.
But once the drug is no longer bound to your binder, then the kinetics are slow enough that you won't have like a majority product of like that open inactive conformation.
Yeah, so the half-life of ring open, ring closed, so going from ring closed to ring open at room temperature is about 2 hours at pH 7.4. At body temperature, it's about 45 minutes or so. It's a little bit higher. What's interesting is that serum albumin tends to prefer to bind the ring open form. So that's also drawing the equilibrium towards inactive. So it can recapture, so the protein could recapture the ring closed form if it unbinds it, I guess I'm not sure if that was your question, but it is in a sort of complicated dance with all the other proteins in the cell. Yeah. Does that answer your question?
I have a follow-up question.
Yeah.
So if you're going to deliver the compound.
Here, I'll give you this, even though this is like a question to me.
If you're going to deliver the protein with the compound with the protein bound and expect it to unbind and bind the actual target, does that mean you need to sort of thread the needle where the binding affinity to the delivery protein is higher than to the target? Because otherwise you're going to sequester too much of it with the delivery protein?
I got to actually steal this back order to answer the question. So I think it's an interesting question, all of this, like what is the optimum, and it all involves, tinkering. But I think that if we really cared about release of the drug and thought that was super important, and we didn't, if the protein gets internalized, so there's a lot of, so the ADC field and delivery field, targeting field, there's, people think that maybe internalization of an antibody drug conjugate isn't that important, but just localizing the drug in the tumor microenvironment is enough, because also the bystander effect, people say, okay, is this real? Is this not real? There's so much debate about what's the most effective way to deliver a drug. It's hard to know. But if we really cared about effective release and like logic gated release, we could design in something that was, for example, pH sensitive, cathepsin proteolizable release, et cetera. We didn't really go so far to do that here, but that is something we could do by design. Okay, I'm gonna pass the baton here.
Sorry, thank you. It's kind of adjacent of what they were asking. Do you, by any chance, analyze the K on and K off, or only the KD that is like the summary of K on and KD?
So did we analyze the kinetics of binding?
Yeah.
Personally, I did not. I don't know if Kaya did, our lab tech who did almost all of these experiments, who I am eternally grateful for. But maybe Nick would know. I don't know.
Yeah, it's a good question. Sorry. No, we have to have the Zoom people here.
That's right, Zoom people. All right. Hey, Ben, really cool talk. I was kind of curious about the results that you showed for Rosetta fold prediction, because supposedly you in your 35 loops that you're using Rosetta fold for prediction, but then you said that Rosetta fold actually has the wrong chirality prediction. Like, why is there a discrepancy there?
Yes, so it turns out that chirality difference is in the exposed part of the ligand, so I don't think it made too much of a difference for our actual design. I think because we had RMSD as part of the filtering for what new backbones went into the LASErMPNN like sequence design process, anything that flipped the other stereo center, which is actually buried, would have been like filtered out that way. But essentially what we've done in like what we've learned from this process is add filters for chirality being predicted correctly, adding a conformer minimization step, which uses Rosetta to constrain to like our DFT conformer instead of letting it just glom up everything and ruin it. But I think we got sort of lucky in this case for our like initial implementation that it didn't mess anything up that was like super essential for the interactions we were designing.
Thanks. Yeah, I guess one related question. For the 35 cycles that you're doing, is it the backbone that's changing or the ligands changing? Like what's actually changing in each of the 35? I might have missed this part.
So I can allude, or I alluded to this earlier, and I apologize for not skipping directly to it, but it's sort of both, right? And it's very subtle, like looking at this from a big picture view, like both things look sort of similar, but you can see like maybe this process has optimized this helix, right, to be a little bit flatter, right? And maybe the ligand has shifted a little bit. But again, these are sort of structure predictions. So it's hard to say without crystal structures of like everything along the way.
Thanks.
Yeah, so for your 35 design loops, if you look at like the LASErMPNN sequences, like how many changes does it make towards the end? Like, or is it like really only changing 2 residues between cycle 30 and 35? If you looked at that?
I don't think we looked at like the generated sequence entropies, but that would be something interesting to look back in retrospect because I have all of that data. But no, we haven't looked at that. Yeah, essentially when I decided to stop the loops was once the distribution of RMSD and like PLDT had sort of converged, and no backbone ligand placements with higher Rosetta fold PLTDT were sampled.
Hi, Ben. Thank you for the talk. Really, really interesting. I guess bringing it back towards the applications conversation, I'm curious, as you think about the potential application of like using these as an ADC-like molecule, a couple questions came up for me in that, is it possible to multimerize these backbones? Like if you wanted to create a multivalent interaction with multiple molecules? And the second question that I've always wondered with like helical bundles like these, how well do they behave in solution? Like in terms of stability and like just performance in that sense? Curious to get your thoughts on that.
So Nick might know more about the latter since he's Mr. 4-helix bundle, but I will give my best answer. And I'm fairly certain that all of these designed 4-helix bundles are like hyper thermostable and like hyperstable generally. And dimerization, multimerization, all of that is absolutely a total like next direction. And like that's the beauty of doing like complete de novo design, right? We have all of the knobs to turn.
Yeah, one thing I'll add to that, since I'm here, is that protein like this, they can be designed to be quite stable, quite soluble, monomeric proteins. They don't bind to anything else. I mean, you might be familiar with these mini protein binders that can maybe bind to protein A and then probably proteins B, C, D, all the proteome, and maybe they're a little sticky, but we don't design hydrophobic residues on the outside of these things, so we don't really design them to bind to anything. You could use these, actually, it's a good point. You could use these as little mini protein binders to other proteins. There's a whole outer surface you could use as a mini protein to actually target itself, as opposed to fuse a targeting protein to it. So they're to answer your question, they're quite well behaved in the sense that they have very stable, high expression yield, soluble, et cetera. You can actually think about binding multiple molecules by simply sort of translating the binding site down, because they're quite symmetric molecules. So you could even just translate it down, extend the protein, bind perhaps multiple copies of the same drug, have multiple binding sites. So you could actually bind more than one molecule with these.
Thank you. Have you run enough stuff yet to figure out if there's any bias in the model, or maybe certain properties of molecules do better than others, polarity, charge?
Yeah, that's a great question. I'm trying to think if there's anything like that stands out to me that I've observed. I would say what I would anticipate, because of sort of the pre-training formulation of how we handle building up the representations for the ligand atoms, right? The model is probably going to be biased or do better towards things that are more small molecule-like, right? So something that I've done is take the ligand-MPNN nucleic acid interaction test set, right, and evaluate the model over that. And what we see is we actually do worse, right? And maybe that's because we're not unfreezing those embeddings and not connecting every single ligand atom to every other ligand atom in order to build up representations of a really big molecule, right? So that's an example of something.
When are you going to try enzymes?
Whenever you're ready. Yeah, I think to give a more serious answer, like this is a great first step, right? In order to do any sort of de novo enzyme design, you need to bind your substrate, right? And you need to optimize specifically binding your transition state, right? So if we can't bind just an arbitrary ligand, we're not going to be able to do enzymatic chemistry on an arbitrary ligand.
So,
Yeah, maybe.
all right, thanks, Ben. So next month we'll be at MIT, I believe, Sergey. Okay, and Ben is here. Stick around and have some more pizza and drinks and chat with Ben about LASErMPNN.